स्टडी विथ मी ब्लॉग्स में आप सभी का स्वागत है। ठीक है, आज हम कोशिश करेंगे 1 आवर का स्टडी मतलब करने का। तो आज हमारा टॉपिक होने वाला है, बेसिकली हम किस बुक से आज पढ़ रहे हैं, गाइटन एंड हॉल टेक्स्ट बुक ऑफ मेडिकल फिजियोलॉजी। ठीक है, एंड चैप्टर नंबर 2 पढ़ रहे हैं, द सेल एंड इट्स फंक्शन। ठीक है, तो सेल एंड इट्स फंक्शन में कुल मिला करके कितना पेज है टोटली हम कैलकुलेट कर देते हैं। उसके बाद थोड़ा पढ़ने को आसानी मिलेगा। 2 3 4 5 6 7, तो 7 मतलब 14 आधे। क्वेश्चन अगर मिलाओगे, हाँ वही 14 हाफ होगा। सो चैप्टर नंबर 2 हो गया, द सेल एंड इट्स फंक्शन। कोशिश करेंगे 1 आवर तक ले जाने की, बाकी देखते हैं। Glossary of terms: cell membrane, a lipid bilayer that envelops the cell within which protein are embedded. The so-called fluid mosaic model, endoplasmic reticulum, a network of tubular and flat vesicular structure in the cytoplasm. The granular endoplasmic reticulum, closely approximated to ribosome, is involved in protein synthesis. The agranular मतलब smooth endoplasmic reticulum helps in lipid synthesis. Golgi body, intracytoplasmic vesicle involved in intracellular secretion and modification of a protein. Nucleus, the control center of a cell containing large quantities of DNA, which are the gene. These genes determine the characteristic of the cell's protein and also control and promote reproduction of the cell itself. Oxidative phosphorylation, the process by which ATP is synthesized in the mitochondria by the respiratory enzyme phagocytosis. Ingestion of a large particle, pinocytosis, ingestion of a minute particle that forms vesicle of extracellular fluid and particulates constituent inside the cell cytoplasm. Each of 30 trillion cells in human beings is a living structure that can survive for months or years, provided its surrounding fluid contain appropriate nutrients. Cells are the building blocks of the body, providing structure for the body tissue and organ. Ingesting nutrients and converting them to energy and performing specialized function. Cells also contain the body's hereditary code that control the substance synthesized by the cell and permit them to make copies of themselves. To understand the function of organ and other structure of the body, it is essential that we first understand the basic organization of the cell and the function of its component part. Organization of the cell. A typical cell as seen by the light microscope is shown in figure 2.1. Its two major parts are the nucleus and cytoplasm. The nucleus is separated from the cytoplasm by a nuclear membrane, and the cytoplasm is separated from the surrounding fluid by a cell membrane, also called as plasma membrane. The different substances that make up the cell are collectively called protoplasm. Protoplasm is composed mainly of 5 basic substances: water, electrolyte, protein, lipids, carbohydrate. The principal fluid medium of the cell is water, which is present in most cells except for fat cell in a concentration of 70% to 85%. Important ions in the cell include potassium, magnesium, phosphate, sulfate, bicarbonate, and smaller quantities of sodium chloride, calcium. These are all discussed in more detail in chapter number 6, which consider the interrelations between intracellular and extracellular fluid protein. After water, the most abundant substance in most cells are protein that normally constitute 10 to 20 percentage of the cell mass. These protein can be divided into two types: structural protein, functional protein. Structural protein are present in cell mainly in the form of long filament that are polymer of many individual protein molecules. A prominent use of such intracellular filament is to form microtubules that provides the cytoskeleton of such cellular organelles as cilia, nerve axon, the mitotic spindle of the cell undergoing mitosis, and a tangled mass of thin filamentous tubules that hold the part of the cytoplasm and a nucleoplasm together in their respective compartment. Fibrillar protein are found outside the cell, especially in the collagen and elastin fiber of connective tissue and in the blood vessel wall, tendon, ligament, and so forth. The functional protein are an entirely different type of a protein and are usually composed of a combination of a few molecules in tubular globular form. These proteins are mainly the enzyme of the cell, and in contrast to the fibrillar protein are often mobile in the cell fluid. Also, many of them are adherent to the membranous structure inside the cell. The enzyme come into direct contact with other substances in the cellular fluid and catalyze specific intracellular chemical reaction. For instance, the chemical reaction that split glucose into its component part and then combine these with oxygen to form carbon dioxide and water while simultaneously providing energy for cellular function are all catalyzed by a series of protein enzymes. Lipid. Lipid are several types of substances that are grouped together because of their common property of being soluble in fat solvent. Especially important lipid are phospholipid and cholesterol, which together constitute only about 2% of the total cell mass. The significance of a phospholipid and cholesterol is that they are mainly insoluble in water and therefore are used to form the cell membrane and intracellular membrane barriers that separate the different cell compartments. In addition, to phospholipid and cholesterol. Some cells contain large quantities of triglyceride, also called neutral fat. In the fat cell, triglyceride often account for as much as 95% of the cell mass. The fat stored in this cell represent the body's main storehouse of energy-giving nutrients that can later be used to provide energy wherever in the body it is needed. Carbohydrate. Carbohydrates have little structural function in the cell except as part of a glycoprotein molecule, but they play a major role in nutrition of the cell. Most human cells do not maintain large storage of a carbohydrate. The amount usually average about 1% of their total mass but increases to as much as 3% in muscle cell and occasionally 6% in liver cell. However, carbohydrate in the form of a dissolved glucose is always present in the surrounding extracellular fluid so that it is readily available to the cell. Also, a small amount of a carbohydrate is stored in the cell in the form of a glycogen, which is an insoluble polymer of a glucose that can be depolymerized and used rapidly to supply the cell's energy need. Physical structure of the cell. The cell contains highly organized physical structure called as intracellular organelles. The physical nature of each organelle is as important as the cell's chemical constituent for the cell function. For instance, without one of the organelle, the mitochondria, more than 95% of the cell's energy released from nutrients would cease immediately. The most important organelles and other structure of the cell are shown in figure 2.2. Membranous structure of the cell. Most organelles of the cell are covered by membranous composed covered by membranes composed primarily of lipid and protein. These membranes include the cell membrane, nuclear membrane, membrane of endoplasmic reticulum, membrane of mitochondria, lysosome, Golgi apparatus. The lipid in the membrane provides a barrier that impedes movement of water and water-soluble substance from one cell from The lipids in the membrane provides a barrier that impedes movement of water and water soluble substance from one cell compartment to another because water is not soluble in lipid. However, protein molecule in the membrane often penetrate all the way through the membrane, thus providing specialized pathway often organized into actual pores for passage of a specific substance through the membrane. Also, many other membrane protein are enzymes that catalyze a multitude of a different chemical reaction discussed here and in subsequent chapter. Now, cell membrane. The cell membrane, also called as plasma membrane, involves the cell and is a thin, pliable, elastic structure only 7.5 से लेकर के 10 nanometer thick. It is composed almost entirely of a protein and a lipid. The approximate composition is protein 55%, phospholipid 25%, cholesterol 13%, other lipid 4%, and carbohydrate 3%. The cell membrane lipid bilayer impedes penetration by water soluble substance. Figure 2.3 shows the structure of the cell membrane. Its basic structure is a lipid bilayer which is a thin double-layered film of a lipid, each layer only one molecule thick that is continuous over the entire cell surface. Inter, inter, interspersed interspersed in this lipid film are large globular protein. The basic lipid bilayer is composed of three main type of lipid: phospholipid, sphingolipid, and cholesterol. Phospholipid are the most abundant of the cell membrane lipid. One end of each phospholipid molecule is soluble in water, that is, it is the hydrophilic. The other end is soluble only in fats, that is, it is hydrophobic. The phosphate end of the phospholipid is hydrophilic and the fatty acid portion is hydrophobic. Because the hydrophobic portion of the phospholipid molecule are repelled by water but are mutually attracted to one another, they have a natural tendency to attach to one another in the middle of the membrane. As shown in figure 2.3, the hydrophilic phosphate portion then constitute the two surfaces of the complete cell membrane in contact with intracellular water on the inside of the membrane and extracellular water on the outside surface. The lipid layer in the middle of the membrane is impermeable to the usual water-soluble substance such as iron, glucose, urea. Conversely, fat-soluble substance such as oxygen, carbon dioxide, alcohol can penetrate this portion of the membrane with ease. Sphingolipid derived from the amino alcohol sphingosine also have hydrophobic and hydrophilic groups and are present in small amount in the cell membrane, especially nerve cell. Complex sphingolipid in the cell membrane are thought to serve several functions, including protection from harmful environmental factor, signal transmission, and as adhesion site for extracellular proteins. The cholesterol molecule in the membrane are also lipid because their steroid nuclei are highly fat soluble. These molecules, in a sense, are dissolved in the bilayer of the membrane. They mainly help determine the degree of permeability or impermeability of the bilayer to water soluble constituent of the body fluids. Cholesterol control much of the fluidity of the membrane as well. Integral and peripheral cell membrane protein. Figure 2.3 also shows globular masses floating in the lipid bilayer. These membrane protein are mainly glycoprotein. This model of the cell membrane is often referred to as fluid mosaic model. There are two types of cell membrane protein: integral protein that protrude all the way through the all the way through the membrane, and peripheral protein that are attached only to one surface of the membrane and do not penetrate all the way through. Many of the integral protein provide structural channel or pores through which water molecules and water-soluble substance, especially ion, can diffuse between the extracellular and intracellular fluid. These protein channels also have selective properties that allows preferential diffusion of some substances over others. Other integral protein act as a carrier protein for transporting substances that otherwise could not penetrate the lipid bilayer. Sometimes these carrier protein even transport substance in the direction opposite to their electrochemical gradient for diffusion, which is called active transport. Still, other act as enzyme. Now, integral membrane protein can also serve as a receptor for water-soluble chemicals such as peptide hormone, which do not easily penetrate the cell membrane. Interaction of a cell membrane receptor with specific ligand that bind to the receptor causes conformational change in the receptor protein. This process in turn enzymatically activates the intracellular part of the protein and induces interaction between the receptor and protein in the cytoplasm that act as a second messenger, relaying the signal from the extracellular part of the receptor to the interior of the cell. In this way, integral protein spanning the cell membrane provide a means of conveying information about the environment to the cell interior. Peripheral protein molecules are often attached to the integral protein. These peripheral protein function almost entirely as enzyme or as controller of transport of substance through the cell membrane, जो कि आपका पोर होता है. The function of a protein are summarized in box 2.1. Membrane carbohydrate, the cell glycocalyx. Membrane carbohydrate occur almost invariably in combination with protein or lipid in the form of a glycoprotein or glycolipid. In fact, most of the integral protein are glycoprotein, and about 1/10 of the membrane lipid molecule are glycolipids. The glyco portion of these molecules almost invariably protrude to the outside of the cell, dangling outward from the cell surface. Many other carbohydrate compounds called proteoglycan, which are mainly carbohydrate substance bound to small protein core, are loosely attached to the outer surface of the cell as well. Thus, the entire outside surface of the cell often has a loose carbohydrate coat called the glycocalyx. The carbohydrate moieties attached to the outer surface of the cell have several important functions. Many of them have a negative electrical charge, which gives most cells an overall negative surface charge that repels other negatively charged objects. The glycocalyx of some cell attaches to the glycocalyx of other cell, thus attaching cell to one another. Many of the carbohydrate act as a receptor substance for the binding hormone such as insulin. When bound, this combination activate attached internal protein that in turn activate a cascade of intracellular enzymes. Some carbohydrate moieties enter into the immune reaction as discussed in chapter number 25, cytoplasm and its organelles. The cytoplasm is filled with both minute and large dispersed particles and organelles. The jelly-like fluid portion of the cytoplasm in which the particles are dispersed is called cytosol and contains mainly dissolved protein, electrolyte, and glucose. Dispersed in the cytoplasm are neutral fat globules, glycogen granules, ribosomes, secretory vesicles, and five especially important organelles: the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosome, peroxisomes. Now coming to endoplasmic reticulum. अब कितना रहेगा? नींद आ रहा है. बाप रे. Endoplasmic reticulum. Figure 2.2 shows a network of tubular and flat vesicular structure in the cytoplasm, which is the endoplasmic reticulum. This organelle help process molecule made by the cell and transport them to their specific destination inside or outside the cell. The tubules and vesicles interconnect. Also, their walls are constructed of a lipid bilayer membrane that contain large amount of a protein similar to the cell membrane. The total surface area of this structure in some cell, the liver cell for instance, can be as much as 30 to 40 times the cell membrane area. The detailed structure of a small portion of endoplasmic reticulum is shown in figure 2.4. The space inside the tubule and the vesicle is filled with endoplasmic matrix, a watery medium that is different from the fluid in the cytosol outside the endoplasmic reticulum. Electron micrographs shows that the space inside the endoplasmic reticulum is connected with the space between the two membrane surface of the nuclear membrane. Substance formed in some part of the cell enter the space of a endoplasmic reticulum and are then directed to other part of the cell. Also, the vast surface area of this reticulum and the multiple enzyme system attached to its membrane provide machinery for a major share of a metabolic function of the cell. Ribosome and a granular endoplasmic reticulum. Attached to the outer surface of a many part of a endoplasmic reticulum are large number of minute granular particles called ribosomes. Where these particles are present, the reticulum is called granular endoplasmic reticulum. The ribosomes are composed of a mixture of RNA and proteins, and they function to synthesize new protein molecules in the cell as discussed later in this chapter and in chapter number 3. Agranular endoplasmic reticulum. Part of the endoplasmic reticulum has no attached ribosome. This part is called the agranular or smooth endoplasmic reticulum. The agranular reticulum function for the synthesis of lipid substances and for other process of the cell promoted by intra-reticular enzymes. Then Golgi apparatus. Golgi apparatus shown in figure 2.5 is closely related to the endoplasmic reticulum. It has membranes similar to those of agranular endoplasmic reticulum. The Golgi apparatus is usually composed of a four or more stacked layer of a thin, flat, enclosed vesicle lying near one side of the nucleus. This apparatus is prominent in secretory cells where it is located on the side of the cell from which the secretory substances are extruded. The Golgi apparatus function in association with the endoplasmic reticulum. As shown in figure 2.5, small transport vesicle, also called as endoplasmic reticulum vesicles, continually pinch up from the endoplasmic reticulum and shortly thereafter fuse with Golgi apparatus. In this way, substance entrapped in the endoplasmic reticulum vesicles are transported from the endoplasmic reticulum to the Golgi apparatus. The transported substances are then processed in the Golgi apparatus to form lysosomes, secretory vesicles, and other cytoplasmic components that are discussed later in this chapter. Lysosomes. Lysosomes shown in figure 2.2 are vesicular organelles that forms by breaking off from Golgi apparatus and then dispersing throughout the cytoplasm. The lysosomes provide an intracellular digestive system that allows the cell to digest damaged cellular structure, food particles that have been ingested by the cell, unwanted matter such as bacteria. The lysosomes is quite different in various cell types, but it is usually 250 से लेकर के 750 nanometer in diameter. It is surrounded by a typical lipid bilayer membrane and is filled with large number of small granules 5 से लेकर के 8 nanometer in diameter which are protein aggregates of as many as 40 different hydrolase मतलब digestive enzyme. A hydrolytic enzyme is capable of splitting an organic compound into two or more parts by combining hydrogen from a water molecule with one part of the compound and combining the hydroxyl portion of water molecule with other part of the compound. For instance, protein is hydrolyzed to form amino acid, glycogen is hydrolyzed to form glucose, lipid are hydrolyzed to form fatty acid and glycerol. ठीक है। Hydrolytic enzymes are slightly concentrated in lysosomes. Ordinarily, the membrane surrounding the lysosome prevents the enclosed hydrolytic enzyme from coming in contact with other substances in the cell and therefore prevents their digestive action. However, some condition of the cell break the membrane of some of the lysosomes, allowing release of the digestive enzyme. These enzymes then split the organic substances with which they come in contact into small, highly diffusible substances such as amino acid and glucose. Some of the specific function of lysosomes are discussed later in the chapter. Peroxisome are similarly physically to lysosomes, but they are different in two important ways. First, they are believed to formed by self-replication or perhaps by budding off from the smooth endoplasmic reticulum rather than from the Golgi apparatus. Second, they contain oxidases rather than hydrolases. Se- Several of the oxidases are capable of combining oxygen with hydrogen ion derived from different intracellular chemicals to form hydrogen peroxide. Hydrogen peroxide is a highly oxidizing substance and is used in association with catalase, another oxidase enzyme present in large quantity in peroxisome, to oxidize many substances that might otherwise be poisonous to the cell. For instance, about half the alcohol a person drinks is detoxified into acetaldehyde by the peroxisome of the liver cell in this manner. A major function of a peroxisome is to catabolize long chain fatty acid. Now, secretory vesicle. One of the important functions of many cells is secretion of special chemical substances. Almost all such secretory substances are formed by the endoplasmic reticulum Golgi apparatus system and are then released from the Golgi apparatus into the cytoplasm in the form of a storage vesicle called as secretory vesicle or secretory granules. Figure 2.6 shows typically secretory vesicles inside pancreatic acinar cell. These vesicles stores protein proenzyme enzyme that are not yet activated, that is known as proenzyme. The proenzyme are secreted later through the outer cell membrane into pancreatic duct and hence into the duodenum, where they become activated and perform digestive function of the food in the intestinal tract. सो अभी थोड़ा नींद लग रहा है, तो यहाँ पर सिक्रेटरी वेसिकल तक यहाँ तक ख़त्म नहीं कर पाया और शायद 4-5 पेज है, तो कुल मिलाकर के 10 पेज और है। 8-10 पेज आई थिंक होगा। सो आज के स्टडी विथ मी ब्लॉग्स में इतना ही। थैंक्स फॉर वॉचिंग।